screencast. This time we'll be discussing the historical experiments surrounding the discovery of DNA as the genetic material. Okay, so let me get my pen, and we're going to be discussing, by the end of this you should be able to discuss in relative detail here, the experimental evidence supporting DNA as the molecule of heredity, as stated by, in this time, Griffith. In subsequent screencasts, so you see that this is screencast 1, and then there will be a 2, a 3, and then a 4 as well. Okay, so this time today we are talking here about Griffith's experiments. Actually, we're going to go back a little bit before Griffith and talk about Friedrich Miescher back in the 1870s, and he's going to characterize the contents of the nucleus. So here's Miescher, and he takes white blood cells. So that's a white blood cell. This one over here is a red blood cell. So these are different types of white blood cells, and you see that these have nuclei in them. And so what Miescher does is he extracts the nucleus, and he determines that the nuclear contents are acidic. Right? Hence, he coins the term nucleic acid. Right? So he is able to determine this, but does not know, of course, that it is DNA by any stretch of the imagination. Right? So Miescher gives us the term here, nucleic acids. Fast forward a few years into the 1920s, and Griffith here discovers what he calls a transforming agent, which can be inherited. So a little bit of background here to understand the details of his experiment and how he figures out this transforming agent. Griffith is working with a couple different strains of the bacterium here, Streptococcus pneumoniae, and there is a rough strain, and then there is a smooth strain described for what they look like here. So the rough strain, when you inject the rough strain into a mouse, Mus muscus, harmless, right? The mouse survives, no problem. So the rough version of strep pneumoniae is harmless, but however, the smooth version of this, the smooth strain, when you inject it into Mus muscus, is what he describes as being virulent, right? And the mice die from that. And so a little gruesome, but these are the experiments that he's conducting back in the 1920s. He knows this, and so this is his model system. Right? And so we will look at scientists, and they have model systems. Right? A system being kind of code word for organism. Right? So the experimental organism of choice here was strep pneumoniae and then Mus muscus. So he's basing off his experiments using this model system. So he goes back, and his experiments are like this. So in experiment 1, he's got this negative control group where he takes, these are harmless, living R, strep pneumoniae here. He injects them into the mice, and the mice live. Right? No problem. This was what he was expecting to see, so he gets all these living mice. He then does a positive control where he takes living S, which again is the harmful one, injects that into the mice, and the mice die, and he knows, okay, I'm dealing with living S, which is harmful, and I'm dealing with living R, which is harmless. So then the next thing that he does, an experimental group, so the line is kind of, I'll draw the line right here. So next thing he does, so these are his control groups up there. The next thing he does is experimental 1. He takes living R, and he kills them using high heat. So these are heat-killed S. Right? So the heat-killed S, he hypothesizes, all right, since I killed the S ones, if I inject those heat-killed S into the mouse is going to live. And sure enough, you get living mice here, right? Or this is what he thinks is going to happen, right? So this sort of is the hypothesis. Right? The next experiment is that he mixes living R, again, which is harmless, and heat-killed S, which is harmless, right, over here. And he predicts that he should get living mice, no problem, right? So when he does this, of course, he gets the opposite of what he's expecting, right? So here's the experimental 2 results, part A. There are several results here. Remember, this was living R, harmless, and this was heat-killed S, which was harmless. And he was hypothesizing that his mouse should live, right? But here he saw, obviously, dead mice. So this makes Griffith go, what is going on here, right? I should have gotten living mice, but I didn't. So he does a little bit more investigating, looks at this mouse, or these mice, and takes samples, tissue samples and fluid samples from the mice here. And another result, right, was that now, when he pulled tissue samples out of there, he saw living S. This really made Griffith sort of step back and say, like, what the heck is going on here, right? I'm getting living S out of this dead mouse when I put in heat-killed S and living R. Right? So the conclusions are what? Well, that the heat-killed S came back to life. Heat-killed S came back to life. Probably not really, a good conclusion to come to. Right? He's never seen dead things come back to life before. But what he did put in there was living R. Right? So his other conclusion, this is what he logically comes to, is that something, and these are his words, transformed the living R into living S. Right? And this is the logical conclusion he comes to. Something transformed the living R into living S. He does not know what this something is, and so therefore he calls this something a transforming agent. This transforming agent. Something transformed the living R into living S. Not only that, but then he goes further. He takes this sample that he extracted from these dead mice with living S in it now, and he injects the living S into subsequent mice, right? And he would predict, based on his positive control, that living S should kill the mice, and of course they do kill the mice. Right? And then what he did with those mice that were killed here is he extracted another sample from that, right, and found more living S. He found no living R in any of these, right? So no living R, right? So whatever that transforming agent was, he was finding that that transforming agent was then passed on to the next generations of these strep pneumoniae. Right? So this transforming agent can be and is passed on generation to generation to generation. Right? This is the definition of hereditary material. Right? This is what the search was now on. All right, what is this hereditary material? Right? So what is this stuff that's able to convert living R cells into living S cells? That's able to change, if you will, not necessarily using this terminology, but what is this stuff that's able to change the phenotype of these cells? Right? So we got this going on over here, right? We're changing the phenotype. What is this stuff that changes the phenotype of cells? This is the question. What is it? Well, unfortunately, Griffith dies before that answer comes to light. So he doesn't know, but it inspires several other experiments that I'll discuss in another screencast of Avery, McCarty, and McLeod, right, and how they discover that it's going to be DNA and not protein. So here is Griffith's summary, a summary slide of his experiments. You should be able to identify the different parts there, what the negative control is, what the positive control is, talk a little bit about his logic and how the evidence then supports his conclusion that there is some transforming agent. They're able to change the phenotype of those cells. Okay? So again, what is the experimental evidence? Okay? How does he know that that DNA, not supporting, he doesn't know that it's DNA yet, right? But that we're getting closer and closer to this, right? So what experimental evidence does he have to getting closer to some molecule as the hereditary, or the molecule of heredity? Okay? So again, that was for Griffith. Right. Thanks for listening, and we'll see you in class.